Hello, I am Pushpangna Singh, a Prime Minister Research Fellow from IIT Kanpur in the May 2020 lateral entry cycle. So in this lecture, I am going to focus on the Orthomyxoviridae, which contains the influenza virus. The family of Orthomyxoviridae is designated by viruses that have a negative sense, single-stranded and segmented RNA genome. The definition of a negative sense RNA virus came from work by David Baltimore, who showed that the package genome of this class of viruses is complementary to the messenger RNA or mRNA, which is designated as positive. There are 6 different genera in the family of Orthomyxoviridae. The influenza viruses A, B and C, Togotovirus, ESA virus and a new genus, Quaranilivirus. Members belonging to any of the 3 different genera of influenza viruses can undergo genetic resortment and thus readily exchange genetic information. However, resortment between members of different genera has never been reported. This absence of genetic exchange between viruses of differing genera is one manifestation of speciation as a result of evolutionary divergence. Different influenza virus strains are named according to their genus, the species from which the virus was isolated, location of the isolate, the number of the isolate, the year of isolation, and in the case of influenza A viruses, the hemagglutinin and neuraminidase subtypes. For example, the 220th isolate of an H5N1 subtype virus isolated from chickens in Hong Kong in 1997 is designated influenza A/chicken/Hong Kong/220/97 H5N1 virus. There are now 17 different hemagglutinin H1 to H17 subtypes and 9 different neuraminidase N1 to N9 subtypes for influenza A viruses as well as a new N10 neuraminidase, while the influenza B virus hemagglutinins and neuraminases are each classified into 2 lineages. Influenza A viruses have a complex structure and possess a lipid membrane derived from the host cell. This envelope harbors the hemagglutinin or HA, the neuraminidase or Na, and the M2 proteins that project from the surface of the virus. The matrix protein M1 lies just beneath the envelope and the core of the virus particle is made up of the RNP or the ribonucleoprotein complex. It consists of the viral RNA segments, the polymerase proteins PB1, that is polymerase basic 1, PB2, polymerase basic 2, and PA, polymerase acid, and the nucleoprotein or NP. The NEP or NS2, which is the nuclear export protein or the non-structural protein 2, is also present in purified viral preparations. The overall composition of virus particles is about 1% RNA, 5 to 8% carbohydrate, 20% lipid, and approximately 70% protein. However, these results will have to be revisited using more quantitative approaches. Specifically, it will be important to get more quantitative data on the presence of individual viral components as well as cellular components that are packaged into the virus. Excellent analysis have already been performed using electron microscopy of negatively stained or frozen hydrated or cryo-electron microscopy particles and tomographic reconstructions. The morphology of influenza A virus particles is characterized by distinctive spikes that are readily observable in electron micrographs of negatively stained virus particles. These spikes, made up of HA and Na, have lengths of 10 to 14 nanometer with an approximate ratio of 4 HA to 1 Na. The influenza viruses are pleomorphic. The spherical particles have a diameter of about 100 nanometer, but filamentous particles with elongated viral structures more than 300 nanometer have frequently been observed, particularly in fresh clinical isolates and in preparations of viruses with specific M1 or M2 proteins. Less is known about the internal structures of influenza viruses. However, the underlying M1 layer can be visualized and reveals a helical superstructure. The RNP complexes were first separated by Deutschberg on sucrose gradients and were visualized by electron microscopy using positive staining with uranyl acetate. These RNP structures appear to consist of a strand that is folded back on itself to form a double helical arrangement. Most recently, attempts have been made to visualize RNPs or individual RNA segments by electron microscopy of thin section virus particles. Influenza B viruses are mostly indistinguishable from the A viruses by electron microscopy. They have 4 proteins inserted in their lipid envelopes: the HA, Na, NB and BM2. The M1 and RMP complexes make up the interior of the particle. It has been shown that the influenza B virus NEP or NS2, which is a non-structural protein 2, is associated with purified virus preparations. Influenza C viruses have been found to possess hexagonal reticular net-like structures on the surface and to form unusually long, that is around 500 millimeter, cord-like structures on the surface of infected cells. Influenza C viruses also contain a core of 3 polymerase proteins and the NP, which are associated with 7 RNA segments. The influenza C virus M1 appears to have a similar role to those of influenza A and B viruses. The major glycoprotein HEF or hemagglutinin esterase fusion combines the functions of the HA and Na, and thus influenza C viruses contain one less RNA segment than do the A and B viruses. The HEF is inserted into the lipid membrane, as is the glycosylated CM2, which is structurally analogous to the M2 of the influenza A viruses and the NB of the influenza B viruses. All A and B type influenza viruses possess 8 RNA segments, whereas influenza C virus only have 7 RNA segments. This was first shown by using a polyacrylamide gel electrophoresis of isolated RNAs from two parent influenza A virus strains and their resultants. Identifying the derivation of an RNA segment by gel electrophoresis is a resultant, and simultaneous protein analysis by serological or gel analysis allowed the assignment of individual RNAs to specific viral proteins. Interestingly, influenza A viruses increase the coding capacity of their genomes by both splicing and uses of alternative open reading frames. The M and NS genes each give rise to a spliced mRNA encoding the M2 and the NEP or NS2 protein. The PB1F2 and PB1N40 proteins are expressed from alternative open reading frames within the PB1 gene. Although not all influenza A virus strains encode these proteins, making them true accessory proteins. Each viral segment contains non-coding regions at both the 5' and 3' ends. The extreme ends are conserved among all segments of influenza A viruses and is followed by a segment-specific non-coding region. The influenza B virus genome is similar to that of influenza A virus. Again, 8 RNA segments code for one or more viral proteins, with the 3 largest RNAs coding for the polymerase proteins, the 4th RNA for the HA, and the 5th and 6th RNAs for the NP and NA respectively. The NA gene codes for the NB protein as well as for the NA. The NB protein is enclosed by a -1 open reading frame 7 nucleotides upstream of the NA coding frame. The 7th RNA codes for the M1 protein, that is around 248 amino acids in length. In termination codon, its termination codon overlaps with its initiation codon for the BM2, which is 109 amino acids in length, which allows for a stop-start translation mechanism. This 8th RNA codes for the NS1 as well as for the NEP NS2 protein, the latter via a spliced mRNA. Cognate PB1F2 and PB1N40 proteins have not yet been identified in influenza B viruses. The non-coding regions of the influenza B virus genome are longer than those in influenza A virus. The genome of influenza C viruses has only 7 RNA segments, with the 3 largest RNAs each coding for one of the polymerase proteins. The PB1 and PB2 proteins are homologous to the corresponding influenza A and B virus proteins. The third influenza C virus polymerase protein is named P3 because it does not display acid charge features at neutral pH as do the corresponding PA proteins of influenza A and B viruses. The 4th RNA codes for the HEF protein, combining the hemagglutinin, receptor destroying and fusion activities. The NP is enclosed by the 5th RNA. The 6th RNA codes for the M protein which is expressed from a spliced mRNA, and from the unspliced mRNA, a long precursor is translated, which is then processed by signal peptide cleavage into CM2. This 115 amino acid long protein consists of an amino terminal extracellular domain with a carbohydrate chain, a hydrophobic transmembrane domain, and an intracellular cytoplasmic tail. Finally, RNA7 codes for the NS1 protein, that is 246 amino acids long, and via spliced mRNA for the NEP-NS2 protein, that is 182 amino acids long. Evolutionarily, influenza A, B, and C viruses have a common precursor, and it is also likely that influenza A and B viruses diverged from each other more recently than influenza C viruses. Based on comparative sequencing studies using the hemagglutinin molecules, it has been postulated that the influenza A viruses diverged about 2000 years ago and the influenza B and C viruses about 4000 and 8000 years ago respectively. Clearly, these numbers are based on a series of unprovable assumptions, including a steady rate of evolution over time, and therefore must be taken cum grano salis. So these are the different important components of the influenza genome. The PB2 is a component of RNA polymerase that is required for cap recognition. The PB1 is also a component of RNA polymerase, but it is required for elongation. The PB1F2 component has a pro-apoptotic activity and it is an IFN antagonist. The function of PB1N40 is unknown. TA is a component of RNA polymerase and it has a protease and endonuclease activity. HA is a surface glycoprotein. It has receptor binding, fusion activity. It is a major antigen and it is involved in assembly and budding. The NP is RNA binding. It is also involved in RNA synthesis and in RNP nuclear import. The Na is a surface glycoprotein along with HA and it has neuraminidase activity. The M1 is a matrix protein and it interacts with RNPs and glycoproteins. It is involved in RNP nuclear export, assembly and budding. The M2 is the membrane protein. It has ion channel activity and it is also involved in assembly and budding. The NS1 or the non-structural 1 protein has different kinds of function, but majorly it has IFN antagonist activity. The NEP or the NS2, which is a non-structural 2 protein, is involved in RNA nuclear export. It is also involved in regulation of RNA synthesis. Influenza viruses bind to neuraminic acid or sialic acids on the surface of cells to initiate infection and replication. The interaction of influenza viruses with a ubiquitous molecule such as sialic acid is constrained by the fact that the HAs of viruses that replicate in different species show specificity towards sialic acid with different linkages. Human viruses preferentially bind to the N-acetyl neuraminic acid attached to the penultimate galactose sugar by an A2 alpha-2,6 linkage, whereas avian viruses mostly bind to sialic acid with an alpha-2,3 linkage. In agreement with this finding is the fact that human trachea epithelial cells contain mostly sialic acid with alpha-2,6 galactose, while the gut epithelium from ducks possess mostly sialic acid from alpha-2,3 galactose sugar moieties. It should be noted, however, that this viral specificity is not absolute and that avian and human cells can contain both neuramic acid linkages, that is 2,3 as well as 2,6. Studies on ciliated cells in the human airway epithelium have shown that sialated proteins with an alpha-2,3 linkages are present and that these cells can be infected with avian influenza viruses. Furthermore, glycan structure is far more complex than just the terminal sialic acid linkage, and evidence suggests that factors such as the type of backbone, chain length, and branching pattern, as well as sulfation and fucosylation, may also influence the interactions with HA. Glycan microarrays that contain a wide spectrum of glycan structures are now being used as tools to better understand the specificity of receptor binding. Also, when viruses are passaged in a particular host, they can adapt to that host by mutating the receptor binding site of the viral HA. In a study of the A/New York/1/1918 virus HA, it was shown that the binding specificity can be changed by a single amino acid mutation, that was D192E, to a preference for alpha-2,3 linked sialic acids. It is thus hypothesized that the HA gene of 1918 influenza virus has its origin in avian species and that a single amino acid change, that is E192D, allowed the hemagglutinin to recognize the alpha-2,6 linked sialic acids prevalent in human cells. While some viruses, for example, the paramyxoviruses and herpes viruses, can enter cells directly through the plasma membrane by a pH-independent fusion process, influenza viruses require a low pH to initiate fusion and are therefore internalized by endocytic compartments. There are four internalization mechanisms: A, via clathrine coated pits; B, via caveoli; C, through non-clathrine non-caveoli pathways; and D, through macropinocytosis. Clathrine-mediated endocytosis has traditionally been the model for influenza virus entry. However, a non-clathrine, non-caveoli mediated internalization mechanism has also been described for influenza viruses. The latter pathway is dependent on low pH and trafficking to late endosomes, as it requires protein kinase C, RAB5 and RAB7 functions. More recently, through the use of specific inhibitors and RNA interference or RNAi, it has been shown that in addition to entering via dynamin dependent clathrine driven pathway, influenza viruses can also enter via dynamin independent pathway that is characteristic of macropinocytosis. Potential differences in the entry pathways in polarized versus non-polarized cells should also be appreciated, as, for example, the actin cytoskeleton appears to be critical for uptake of influenza viruses into polarized cells but not non-polarized cells. The requirement of specific host proteins during influenza virus entry will help to further delineate the cellular pathways utilized by the virus. Genome-wise RNA-I screens have identified multiple factors that are required for efficient entry mediated by the influenza virus glycoproteins. However, the ability of the virus to enter via different routes means that this approach is unlikely to capture factors that are specific to one endocytic route. A more focused approach, such as that which shows the requirement of epsin-1 for clathrin-mediated uptake of influenza virus, is needed. The epidermal growth factor receptor, or EGFR, has also been demonstrated to play a role during influenza virus entry, and it is thought that virus attachment to the cell stimulates EGFR, which leads to activation of signaling cascades such as phosphatidylinositol 3 kinase or PI3K signaling, which is known to promote influenza virus entry. There are also questions over whether sialic acid is the only attachment molecule. Leg-1 cells, which are deficient in N-linked glycosylation but still contain sialylated proteins, are unable to internalize influenza viruses. Similarly, cells deficient in sialic acid can be made to support influenza virus entry if they express one of the C-type lectins, DC sign or L sign. Influenza viruses and other envelope viruses including rhabdo, lavi, bunya and eloviruses require low pH to fuse with the endosomal membranes. After binding to the target cell, the surface and endocytosis, the low pH of the endosome activates fusion of the viral membrane with that of the endosome. This fusion activity is induced by a structural change in the HA of influenza viruses, but in order for this to occur, the HA knot precursor must first be cleaved into two subunits, HA1 and HA2. Once in the acid environment of the endosome, the cleaved HA molecule undergoes a conformational change, and this exposes the fusion peptide at the end terminus of the HA2 subunit, enabling it to interact with the membrane of the endosome. The transmembrane domain of the HA2 inserted into the viral membrane and the fusion peptide inserted into the endosomal membrane are in juxtaposition in the low pH induced HA structure. The concerted structural change of several hemagglutinin molecules then opens up a pore which releases the contents of the virion, that is viral RNPs, into the cytoplasm of the cell. The precise timing and location of uncoating, for example maturity of the endosome, depends on the pH mediated transition of the specific HA molecules involved. The uncoating of influenza viruses in endosomes is blocked by changes in pH caused by weak bases. For example, ammonium chloride or chloroquine, or ionophores, for example, monensin. Effective uncoating is also dependent on the presence of the M2 protein, which has ion channel activity. Early on, it was recognized that amantadine and rimantadine inhibit replication immediately following virus infection. Later, it was found that the virus-associated M2 protein allows the influx of H+ ions from the endosome into the virus particle, which disrupts protein-protein interactions and results in the release of RNP free of the M1 protein. Amantadine and rimantadine have been shown to block the ion channel activity of M2 protein and thus uncoating. The HA-mediated fusion of the viral membrane with the endosomal membrane and the M2-mediated release of the RNP result in appearance of free RNP complexes in the cytoplasm. This completes the uncoating process. The time frame for uncoating process was examined by inhibiting virus penetration with ammonium chloride. The majority of virus particles showed a half time for penetration of about 25 minutes after adsorption. Barely 10 minutes later, half time of 34 minutes after adsorption, RNP complexes are found in the nucleus. The process for uptake of RNP molecules through nuclear pores is an active one involving the nucleocytoplasmic trafficking machinery of the host cell. Much less is known about the uptake and uncoating of influenza B and C viruses. Influenza B viruses are more like influenza A viruses as both recognize N-acetylnuraminic acid and as receptors, while influenza C viruses bind to 9-O-acetylated nuraminic acid derivatives. Like influenza A viruses, the B and C type viruses go through an endosome-mediated uncoating process which requires proteolytic activation or cleavage of the HA or HEF proteins and subsequent fusion of the virus and endosomal membranes. Although the viral glycoprotein of both viruses are independent on a low pH triggered fusion, the influenza C virus HEF mediated fusion or uncoating may occur at a higher pH in early endosomes. At this point, the role of CM2 in uncoating of influenza C viruses is less well established than that of the BM2 protein of influenza B viruses, which is the homologue of the influenza A virus M2 protein. Much is known about the HA molecule and excellent reviews are available. In fact, the influenza virus HA has become a model for studies of protein folding and trafficking, a protein quality control, membrane fusion, protein receptor interactions, and antigen-antibody complexes, and last but not the least, for investigating how the immune system reacts to foreign proteins. The major functions of the HA are the receptor binding and fusion activities, but there may also be a structural role for the HA in budding and particle formation. The HA is trimeric rod-shaped molecule with the carboxy terminus inserted into the viral membrane and the hydrophilic end projecting as a spike away from the viral surface, that is, a type 1 integral membrane protein. Early on, it was shown that post-translational modification of the precursor molecule, that is glycosylation and palmitolylation, cleavage of the signal peptide in the endoplasmic reticulum and ER, and cleavage of the HA0 precursor into HA1 and HA2 subunits are required for the full activity of the molecule. The first X-ray crystallographic structure of an HA, the ectodomain released from the virus by bromelain treatment, was resolved in 1981 by Ian Wilson. At that time, the HA from the H3N2 virus was the largest biological molecule for which a structure had been solved, and it started an unprecedented drive to study the structure-function relationships of biologically important molecules, which continues unabated to this day. The structures of numerous HAs have now been resolved, including the subtype 1 HA molecule of the 1918 and 2009 pandemic influenza viruses and that of an H2 subtype HA. Remarkably, Even though the overall amino acid sequence identity can be less than 50%, the structure and functions of these HAs are highly conserved. This represents a case of evolution and sequence variation proceeding to an extreme level while structure and function have remained conserved. Even more surprising is that the structure of the influenza B virus HA is similar to that of influenza A virus HA despite sharing only 25% sequence identity. The crystallographic structure of the uncleaved influenza A virus HA is superimposable onto that of the cleaved HA1 and HA2 with the exception of the amino acids adjacent to the cleavage site. The major features of the structure are a long fibrous stem which is made up of triple stranded coil of alpha helices derived from the three HA2 parts of the molecule, helix A and helix B, and B, the globular head which is made up of three identical domains whose sequences are derived from the HA1 portions of the three monomers. The first major function of HA is binding to receptors, and the receptor binding site lies within the globular head of the molecule. This site has been designated through crystallization and structural analysis of HA receptor complexes as well as by mutational analysis. In the HA subtype viruses, it appears that within the receptor binding pocket of an avian HA, a glutamine in the position 226 preferentially accommodates the 2,3 linked sialic acid, whereas a leucine in that position in human H3HAs preferentially accommodates the alpha 2,6 linked sialic acid. The second major function of HA is acid pH triggered fusion, which is required for the uncoating process. Low pH treatment changes the structure of the HA dramatically. The molecule becomes susceptible to protease digestion, and the disulfide bond linking the HA1 and HA2 subunits becomes susceptible to mercaptoethanol. However, the important feature of the acid pH mediated change is that the fusion peptide becomes aligned antiparallel to the membrane anchor of the HA2. The end result is that the fusion peptide brings the endosomal membrane into juxtaposition with the viral membrane, leading to fusion. The presence of more than one hemagglutinin then leads to the formation of a fusion pore through which the RNP can enter the cytoplasm. Structures of the prefusion HA as well as an early fusion intermediate have helped to reveal the molecular details of the changes that occur during the transition from pre to post-fusion state. Structures of the fusion peptide in lipid environments have been resolved by nuclear magnetic resonance or NMR and show that the peptide forms a tight helical hairpin structure that angles back on itself. This hook structure may help to pull the endosomal membrane close to the viral membrane, resulting in the initiation of the actual fusion process. These studies represent the first foray into the characterization of the transmembrane region, which so far has proved difficult to analyze. The third major structural element of the HA, the cytoplasmic tail, is highly conserved among all subtypes. There are three cysteines that are palmitoylated, with one of them located in the transmembrane domain. The role of the cytoplasmic tail and palmitates attached to the cysteines is not entirely clear due to subtype and cell host specific differences. In addition to having an important role in receptor binding, fusion, and assembly, the influenza virus HA is also the major determinant recognized by the adaptive immune system of the host. Following infection and replication, a vigorous immune response is induced, which usually results in the formation of neutralizing antibodies. These antibodies then lead to the selection of antibody escape variants. The amino acids undergoing changes are almost exclusively on the HA1 and on the outside of the molecule. Many of these changes get fixed, that is, they accumulate over time, designating the antigenic drift of influenza viruses. Fab antibody fragments have been shown to bind to different regions of the HA1, and interestingly, not in all cases do 3 Fabs bind to one trimeric spike. That is in 3:1 ratio. Examples have been found where only one Fab molecule binds to one HA spike, that is in 1:1 ratio, or where just 2 Fab molecules bind to a trimeric spike, or the 2:1 ratio. In the latter case, the two Fab fragments cross-link the two monomers so that the HA molecule cannot undergo an acid pH-induced conformation change. Attempts have also been made to measure antigenic evolution of influenza virus strains by pairwise comparison of hemagglutinin inhibition assays. Unexpectedly, broadly cross-reactive monoclonal antibodies have been identified which do not bind to the tip of the HA molecule but also have neutralizing activity. These antibodies are directed against the conserved stem region of the HA spike and recognize the membrane proximal part of HA1 in combination with HA2 or the HA2 alone. In general, these antibodies recognize the HAs within either group, but recently a human monoclonal antibody was described that binds to the stem of both group 1 and group 2 HAs. The most likely mechanism by which these cross-reactive anti-stem antibodies neutralize influenza viruses is by blocking conformational rearrangements associated with membrane fusion. A broadly neutralizing human monoclonal antibody that recognizes the highly conserved sialic acid binding site of H1HAs has also been described. It is hoped that the conserved epitopes identified by these cross-protective antibodies could be utilized as immunogens with the possibility of developing novel vaccine constructs that will result in effective and safe universal influenza virus vaccines. The M2 protein of influenza A viruses is tetrameric type, that is, it lacks a signal peptide sequence, integral membrane protein. It has a short ectodomain, a transmembrane domain, and a cytoplasmic domain with palmitate and phosphate modifications. M2 has been shown to possess ion channel activity, and its major role is thought to be that of conducting protons from the acidified endosomes into the interior of the virus to dissociate the RNP complex from the rest of the viral components, thus facilitating the uncoating process. The structural and genetic analysis of M2 protein has revealed that the ion channel is acid gated but not voltage gated and highly selective for H plus ions. The structures of the transmembrane regions of M2 and those that included the cytoplasmic sequences reveal a good understanding of the mechanism of protein conductance, which is controlled by the histidine 37 and tryptophan 41 cluster. The transmembrane region, when viewed from the top, shows 4 helices that sit at an angle of the lipid bilayer, forming a pore, which is shown here in this image. As you can see here, these are the 4 transmembrane helices, and these are sitting here at an angle in the lipid bilayer, and this is the pore. Structural studies on M2 in complex with adamantine drugs indicates two potential sites of interaction. In the X-ray structure, a single drug molecule binds to the core of the pore. In contrast, the NMR structure shows four drug molecules binding to the lipid-exposed surface of the channel close to the cytoplasmic ends of the helices. More recent data confirm the existence of these two interaction sites and propose that both drug binding mechanisms may have physiological significance. The structure and precise function of the extracellular portion of the M2 protein remain to be resolved. This external portion of M2 has been considered as the basis of a universal influenza vaccine approach because the M2 protein maintains a highly conserved sequence over long periods of time. The ion channel activity of M2 has also been implicated in stabilizing HAs from premature low pH functions in the trans-Golgi network, but this second function may only come into play for viruses carrying highly sensitive HAs. This is the case for H5 and H7 HAs, which have a multi-basic cleavage site that can be cleaved by ubiquitous proteases and are therefore more susceptible to a premature low pH induced conformational change. Further functions attributed to the M2 protein include roles in particle morphology, genome packaging, membrane scission, and inhibition of autophagy. So this was a lecture on a basic introduction to influenza virus, and we also discussed the other important proteins of the virus, the genome structure. If you have any doubts or any questions, you are most welcome to shoot your question in the chat box. Thank you.